Alright, so today I'm going to be talking about DNA duplication. Here's a little strand of DNA for you. And so, as we all know, these are just two strands running and forming a helix. So here are those two strands. And they're running anti-parallel to each other. So there's two sides to each strand. It just kind of depends on the sugar that's on the end. But anyway, one of them is the 3' end, and one of them is the 5' end. And since they're anti-parallel, the other way around it must be this one's the 3' end, and this would be the 5' end. And so in order to split this DNA, or you have to split the DNA in order to duplicate it. So you have two enzymes that help you do that. Okay, so don't forget, up here you have all these bases, binding together, as you see right here. And so in order to split your DNA like this, there's two important enzymes. One of them is helicase right here, and what that does is it just unwinds the DNA helix and it forms a replication fork right here. And that's so you can replicate the template strands. And then here you have topoisomerase, which basically it just removes twists and knots that form in the DNA so it can be unwound by the helicase. And so we're going to say that they're both traveling in this direction. And so in order to keep these from coming back together, because we just split them apart but they're attracted, so they want to just come back together, there's these proteins called single strand binding proteins, and they're bound to the bases. Like this. And so that they keep the strands from coming back together. They keep it open so you can copy the DNA, basically. So we see that this strand right here is moving from the 3 to 5 direction, just like the helicase and topoisomerase. This one is called the leading strand because you can make one continuous strand of replicated DNA. And then this one down here is the lagging strand because it isn't, like when you're making the DNA, it isn't continuous. And so how does this work? You have primase, which is an enzyme that lays down primer, which is basically just a short section of RNA nucleotides right here. And then DNA polymerase is another enzyme right here. And it attaches to the RNA primers and adds DNA nucleotides to the complement strand. So then it moves in this direction and you get DNA right here. So you see how all this DNA was created in one continuous strand. Well, the lagging strand works a little bit differently. It's still the same primer and polymerase and DNA working, but you have to do it in short fragments called Okazaki fragments. So it's going to be like this. And they're the same, it's the same thing as the leading strand, because it has to assemble in the 3 to 5' direction. So it assembles in these small little fragments called Okazaki fragments. And what happens after that is the fragments are joined together by DNA ligase, which is like basically the glue of DNA. And now you have each side of the DNA copied. And you wind up with, you finish with, there's your template strand. And then you have the two complement strands here that you've just formed, and you've replicated the DNA and you have 2 copies of it now.